Hello, everybody. We are here to discuss on the topic on metabolism deficiency. And the syndrome we chose for today is called Pearson syndrome. We are from group matrix number 206031.
Hi, my name is Anati Nabilah binti TAJUDIN SHAH, and my matric number is 205706.
My name is ZARIFAH binti AZAMUDDIN. My matric number is 207809.
So on the top, what is Pearson syndrome? Pearson syndrome is a rare disorder of mitochondrial metabolism presenting in infancy. It is usually immediately seen in infancy and usually does not prolong until adult. It could be detected easily during infancy, where Pearson syndrome is caused by a single or large deletion of the mitochondrial DNA, which can range from 1,000 to 10,000 DNA building blocks or nucleotides. So the most common deletion which occurs is in about 20% of the affected individuals, usually removes up to 4997 nucleotides. So in summary, Pearson syndrome is basically when the mitochondrial DNA gets deleted and it affects the metabolism in the body. Okay, so the Pearson syndrome, it is also known as Pearson marrow pancreas syndrome, which means this Pearson syndrome, the deletion of the mitochondrial DNA, bone marrow, and also the pancreas. It causes problem with the development of blood-forming hematopoietic cells in the bone marrow that have the potential to develop into different type of blood cells. So that means blood cells that develop in the bone marrow of a person suffering from Pearson syndrome is very different from the regular blood cells that are being produced. So for this reason, Pearson syndrome is considered a bone marrow failure disorder, and it can also affect the function of the pancreas and other organs as well. Okay, who was the first person to discover Pearson syndrome? This person was a pediatric hematologist and an oncologist, Howard Pearson, in 1979, where he was the first one to discover something called Pearson syndrome. But discovering that it is caused by the deletion of mitochondrial DNA was then discovered a decade later. So basically, how does Pearson describe this Pearson syndrome is that it's described as a new syndrome of refractory sideroblastic anemia with vacuolization of marrow precursors and exocrine pancreatic dysfunction. So which means it affects it acts as a marrow precursor, and it also contributes to the pancreatic dysfunction. Okay, so as you can see in the image, those are red blood cells, but the ones that the arrow are pointing is the red blood cells that are produced by the bone marrow from a person who is suffering from Pearson syndrome. Because you can see it's abnormal from the normal blood cell. It does not have a loop. All right.
Okay, so what causes Pearson syndrome? As we all know, Pearson syndrome is a rare fatal disease that present during the infancy. Death of the patients with Pearson syndrome usually occurs early in life before the age of 4 years old. It is caused by mutation in the mitochondrial DNA where one or multiple anti-DNA deletions of varying sizes, location, and proportion occurs in the cell. Most cases of Pearson syndrome occurs by mistake, or also known as de novo mutation, during the early development of the embryo. This indicates that it is not inherited or passed down from the parents. However, there are also rare cases of Pearson syndrome that is caused by germline mutations. A germline variant occurs in a sperm cell or an egg cell and is passed directly from the parent to a child during the fertilization process. Okay, next slide, please, KAVEE. Okay, so in normal mitochondrial DNA, it converts chemical energy from food such as carbohydrates and fats in the form of ATP, or also known as energy, which can be used by the cells for other metabolic pathways. The molecular defect in this disease was initially identified by the deletions of 4,977 base pair in length of mtDNA. Most of the base pair that was deleted in the mtDNA encompass the portions for genetic coding of NADH dehydrogenase, cytochrome oxidase, and ATPase. This mutation affects the electron transport chain in the mitochondria, which results in defective energy production and development of the clinical features. The severity of this disease depends on the degree of heteroplasmy, which is the amount of mutant mtDNA in the cells. In normal tissues, all mtDNA molecules are identical, which is known as homoplasmy. For heteroplasmy, deleterious mutations in the mtDNA coexist with the normal mitochondrial DNAs, where it usually affects some of the mitochondrial DNAs, but not all within the cell or tissue of an individual. Next slide, please. DNA contains genes with specific genetic codes used to produce proteins, and many proteins coded by the mitochondrial DNA are needed to make energy. As the gene coding for the protein complex of the NADH dehydrogenase, cytochrome oxidase, and ATPase, which is important for the ETC, is deleted, the electron transport chain and the ATP synthase cannot function properly to synthesize ATPs for the use in other metabolic pathways. This is due to the impairment of the mitochondrial oxidative phosphorylation, which results in reduced energy production of patients with Pearson syndrome. Most patients die early in the life due to the metabolic acidosis, and those who survive and recover from the hematological manifestation may develop clinical manifestation of patient with Kearns-Sayre syndrome. Next, we will proceed with Rifa to discuss about symptoms and treatments of Pearson syndrome.
Okay, so now we move to Pearson syndrome. The symptom of Pearson syndromes usually affect the cells in bone marrow, which include the red blood cells, white blood cells, and platelets. All right. Some children with Pearson syndromes usually have problem with all these three blood cell types, but other may not, because they may get the same symptoms, same syndromes, but they probably get different symptoms. So sometimes the child that have the Pearson syndromes may also have the pancreas disease, which is cause the fatigue and diabetes, and also hard to gain weight. So here are the symptoms that we can see from the child that have the Pearson syndrome, which is the body become weaker, skin pale, fatigue, easily catch colds and sickness. So the body take longer time to stop the bleeding than compared to normal children, and frequently get diarrhea and stomach pain, and also have other problems with other organs, which is liver, kidneys, heart, eyes, ear, and or brain. Next slide, KAVEE. Okay, so here the way we diagnose the children that have the Pearson syndrome. First, we do bone marrow biopsy, which is to look for signs of sideroblastic anemia or bowel movement sample to measure the amount of fat in the stool. Next, urine test to check the certain organic acids, which will be a sign of metabolic acidosis. Next, genetic testing for change to see the change or mutations in mitochondrial DNA. So this genetic testing may be especially important because usually the person that have Pearson syndrome is caused by deletion of mitochondrial DNA or duplication of mitochondrial DNA. So by diagnosis with genetic testing, the doctor will know whether the condition is caused by deletion or duplication of DNA. Next slide, KAVEE. So here the treatment for the Pearson syndrome disease. Actually, there is no cure for Pearson syndrome. However, the goal of the treatment is to decrease the seriousness of the symptoms so that the child can live healthy and as long of a life as possible. First, the children must get frequent blood transfusion to help supply the body with healthy red blood cells. Next, using the pancreatic enzyme replacement to help replace the missing enzymes needed to digest the food, or insulin injections may be necessary to treat the diabetes. The last one is avoid other people who are sick or viral or bacterial infections, which these children cannot fight off illness as well as other children can. So other treatment depends on the specific symptoms presented by each person with Pearson syndrome. It may be necessary to see specialists for the liver, kidneys, heart, and pancreas organ disease affected. Next slide, KAVEE.
Resume.
Okay, next, prognosis. So for the prognosis of actually not Pearson syndrome usually causes the baby to die once past infancy. He or she may begin to have a fewer symptoms of Pearson syndromes, and the symptoms may be less severe than other children. Okay, many of children who survive past age 4 go on to have symptoms of different diseases called SHREE syndromes. So this syndromes is more likely to affect the brain, spinal cord, and nerve, and the symptoms include weak eyes, muscles, and difficulty coordinating movements. So it is important for the children to be monitored by the doctors for any changes in the symptoms as they get older. So next, I will pass to KAVEE. KAVEE, next slide.
Thank you. Thank you. So, right, okay. So I'm going to give you all some example of past patients, because if you see, Pearson syndrome is actually a very, very rare syndrome where there's only been less than 100 cases that have been reported worldwide. So, there was once where in 1990 it was reported that 5 patients who were diagnosed with Pearson syndrome, Pearson marrow pancreas syndrome, who presented permanent metabolic acidosis and increased ketone body or pyruvate molar ratios in plasma. Another case was reported where it was described syndrome and characterized by sideroblastic anemia, mild pancreatic insufficiency, and progressive syndrome has different effect on different people. Some people are presented with permanent metabolic acidosis. Some people are presented with progressive muscle weakness. So it shows different symptoms on different people as reported in these cases. And there were also 2 children with a thing where they had pancreatic dysfunction. Another two children that was reported with splenic atrophy. 2 patients died at the age of 26 or 29 months. And some even lived up to 36 or 42 months that showed progressed to the treatments, which showed a hematologic improvement. So that is all from all of us. Thank you.